Thank you very much for the invitation, and it is an honor for me to speak to you on this great symposium honoring Sérgio Mascarenhas. I will be talking about multimodality neuromonitoring in neurocritical care. This is one of the areas that I've been working on since I moved back to the Johns Hopkins Medicine 5 years ago. And so these are the objectives of my talk. I will review the conventional paradigm. Then we will be moving on to the more contemporary paradigm of precision medicine, and then describe the aims of our Precision Medicine Center of Excellence, and learn some of potential benefits of precision medicine and multimodality neuromonitoring in neurocritical care. Now, what is the conventional paradigm? This is the paradigm that we've been using for the past couple of decades. What we're trying to do is trying to use multimodality neuromonitoring so that we can provide goal-directed therapy for patients, right? But we're still looking at patients as groups of patients, not individuals. So this is probably the earliest example that we have, in neurocare. This is from the Cambridge. They were probably one of the first, if not the first, to bring in engineers to the neurocritical unit. As you can see here, we have several, this is the unit at Addenbrooke Hospital, and they have several devices. And it's clear that, yes, these patients are complicated. They require not only systemic monitoring, but also intracranial monitoring. But one of the problems is that these devices don't talk to each other. So they're completely disconnected from each other, which maybe it is impeding our progress, right? So they obviously, with the help of Marek Czosnica, who's an engineer, they were able to come up with what we call the PRX, which is the reactivity index here in green. And they were able to derive that based on the correlation between the blood pressure in red, continuous blood pressure monitoring, and cerebral blood flow changes in blue, right? So this was something that it was different. It was something new and allowed us to look at cerebellar regulation in a different light. But still, this requires a lot of computational analysis, and still we have to look at the monitors to actually determine what the other variables are. So it is a big progress, but it's still not ideal. So then this takes us to the concept of precision medicine. So what is precision medicine? And I think obviously there are two concepts that are currently en vogue in medicine. One of them is the so-called comparative effectiveness research, which essentially it will allow us to look at the evidence of benefit to patients when these interventions are administered by us in the ICU or in the hospital in routine clinical practice settings, right? So this is a new concept too that has been gaining momentum. But also the precision medicine one is another one, and it's also called personalized or stratified or P4 medicine. And the idea is to tailor some of the measures or medical treatments that we have to the characteristics of each individual patient, and maybe tailored to that patient also based on each day different characteristics, right? So what that means is that for patient X, treatment Y may be applied, but for patient T, treatment S may be applied, but not treatment X, right? So that would be something that we're working on, and it sounds easier said than done. It's actually very complicated, as I'm going to show you. So and these are the proposed frameworks in general, for clinical research. The first question that we try to answer is whether something could potentially work. And for this, we design randomized controlled trials, right? There are very specific population with inclusion and exclusion criteria, and then there is a treatment, and then we look at an outcome. And then in yellow, we have the question as to whether something could potentially work, and this is when we do the comparative effectiveness research. And then in green is whether something is worth doing, right? Whether it would benefit our patients, it would benefit their quality of life, their productivity, their preferences. And many times we don't ask these questions, but we have to start asking those questions in earnest in neurocritical care. So what does precision medicine in neurocritical care mean to me? To me, it just means to improve the classification of our patients, right? So we need to move beyond the broad, crude classification of mild, moderate, and severe, right? So not all severe TBIs are the same, and not all severe subarachnoid hemorrhages are the same. So essentially what we're trying to do here is trying to change the taxonomy of our we need to apply the right treatment to the right patient. So no more one size fits all. So we have a big group of patients that receive the same treatment. What we're trying to think about with this is trying to apply one specific treatment to one patient, but not to other patients, depending on that patient's characteristics, right? But of course, we have significant challenges, in neurocritical care. One of them is, of course, the impact of acute neurological disorders and injuries on outcome and disability worldwide. It's one of the leading causes of disability, right? The other problem is that we have made advances, but yet the current management has still serious limitations. And we still have significant unmet need for more effective neurocritical care, right? So, and these are more or less, in my mind, the unmet needs in our field. One is, of course, we need to be more specific about detection and diagnosis, right? We need to have more meaningful classification. We have to have better mechanistic targets for therapy. Without them, we won't be able to provide effective treatments. We also need to do better on our prediction modeling, right? So we need to be more accurate. We need higher levels of long-term recovery. And of course, there is also the issue of proportionality of care and outcomes. So there's a lot of unmet needs, which is good in the sense that we have tremendous amount of research to do over the coming years, but it's also a challenge because we're not there yet. One of the best examples that I can give you is this paper that was published by our colleague Jan Klassen from Columbia University that was published in the New England Journal of Medicine in 2019. What Jan was trying to do was trying to classify their patients in coma or clinically in coma in a much better way. So what he did, he used obviously continuous EEG monitoring. He quantitated the EEG, and then using artificial intelligence and neural networks, he was able to actually use two simple paradigms. One of them is asking patients to keep opening and closing in their right hand or their left hand, and then telling them to stop opening or closing their right or left hand. Now, all these patients clinically looked like they were in coma. But once they did the analysis of the quantitative EEG, they realized that 15% of those patients, even though clinically they were in coma, their EEGs was actually reactive. And what that meant is that those patients were more likely to survive and they're more likely to wake up long-term and have a good recovery long-term. So this is one of the best examples, if not the best, as to why we need to do better with the classification of our patients. Same issue goes, of course, is more important for prognostication. As you can see here, looking at all the different disorders that we treat in the neurocritical care unit, yes, we've become much better at predicting, but it's not ideal, right? So if you can imagine, if you put yourselves in the shoes of patients or families, they want us to be very precise, right? They want us to make sure that when we tell them that their loved one is not going to do well, they want to make sure that we tell them that we're 100% certain. And as you can see, we're far from that. So we're not close to meeting that goal. So this is what we need, more precision. And then the same issue goes for our clinical trials. All our clinical trials are failing in the sense that all the therapies that we have investigated in traumatic brain injury, intracerebral hemorrhage, and subarachnoid hemorrhage have given us neutral results. And part of this issue is that we're trying to give the same treatment to all patients without any further classification or improvement of the taxonomy of our patients. Well, the question is obviously whether we could do better, right? Of course we could do better. This is like the Brazilian soccer team, right? So yes, they can always do better. And and one of the ways that we have to do better is to overcome these two big challenges, right? One of them is the challenge of dimensionality and also the curse of heterogeneity, right? I'll tell you a little bit about that. Yes, and what we mean by dimensionality is that the nervous system has a high dimensional process, right? So, and this is a big challenge, right? Because we have all the tremendous amount of complex network of neurons, axons, and dendrites that are all working to send signals to uh, for all the processing, and then you have an output that is complicated already. And then there's also the issue of time, right? So I think we need to understand a little bit more how this all work and apply more complex, sophisticated computational modeling to answer that question. There's also the issue of the challenge of heterogeneity, as I mentioned before. And by that we mean heterogeneity in gene expression, heterogeneity in biological systems, heterogeneity in disease susceptibility. As you can see, not everybody will respond the same to specific treatment. There's the issue of heterogeneity in treatment effect. Not all patients respond the same. And of course, heterogeneity in phenotype and disease trajectories. And I think these are the challenges that we need to address as well. So, and of course, precision medicine is still a provocative hypothesis, right? So, and in this case, we're trying to use multimodality neuromonitoring to address the issue of precision medicine, right? So, precision medicine, of course, is telling us that medicine and health can be more effective and efficient if they're individualized. So, this is a very different concept. And it's also telling us that we could achieve this by leveraging data science to achieve precision and personalization in medicine. So, and of course, we have to think about the building blocks of this process. One of them, of course, is data. And this is where multimodality neuromonitoring comes into place, right? We need to provide the data using all the devices that we have available and more. And then we have to come up with biological hypotheses. And finally, the third block is to come up with robust analytics. So for this, we need, of course, the help of data scientists and biomedical engineers to help us answer our questions. Without their help, we won't be able to do this. The other concept, of course, is the prediction, right? So the concept of prediction. So what is it that we're trying to predict? And I think that's important. We're trying to define that very clearly. Are we trying to predict the state, like in the case of the manuscript that I just showed you by Jan Klaassen from Columbia University, that we're trying to define whether patients were in a cognitive moderate dissociation? Or are we trying to predict events, like seizures or coma or outcomes? Or are we trying to predict treatment response, like TPA response in patients with stroke? So I think it's also important to define what is it that we're trying to predict. There is also the paradigm, the endotype paradigm, which is essentially the same concept that I mentioned to you before, which is the reclassification of disease using an extended repertoire of features, right? So, and this may or may not be genetically mediated. It may be linked to biological mechanisms, to outcome, or to treatment response. And I think we need to define these endotypes for our patients and for each specific disorder, depending on those features that we mentioned already. So for this, we have designed what we call the Precision Center of Excellence in Neurocritical Care, which essentially means is bring in a lot of collaborators to work together to try to. What we're trying to do with this Precision Medicine Center of Excellence, and we also call them the PIMCO, is not only to look at the science per se, but also to look at the clinical care impact, meaning what's the value of what we're doing for our patients, value to healthcare systems, value to insurance companies, value to governments. So what we're trying to do with this is obviously trying to lower not only the cost of care for our patients, but also the barriers to research. So it's not just the scientific piece, but we also have to balance the clinical care impact that our project is actually do, right? So this is our vision. Our vision is to leverage high-resolution, multidimensional data to tackle what we talked about before, come up with mechanistic hypotheses and advanced modeling, including artificial intelligence, but not just artificial intelligence, to effectively and efficiently enhance the care and outcome of critically ill neurological patients. So we knew that in order to get to where we are today, we needed to do several steps. We needed to meet several steps that they were required. One of them was to collect and archive high-resolution physiological data. Then the next step was to integrate that data and also to have high-resolution time synchronization of the data, and to also think about the artifacts of all the signals that we were collecting so that we could identify them and remove them. We also knew that we needed to build a comprehensive database not only including the relevant clinical observations, but also laboratory results, and more importantly, imaging data, right? So by imaging, I mean MRIs and CT scans and all the images that we obtained from the patients. And we were not interested in archiving all those images per se. What we were trying to do was trying to work with our colleagues and biomedical engineers so that we could segment those images and quantify them, and then use the output of the quantification analysis to include in the database. And that's what we're doing. And of course, when I started building this program, we had the equipment that was in the top of the slide that you can see here. We had ways of measuring brain oxygenation, intracranial pressure, also ways to measure continuous EEG, and we had an electronic health record system that is called EPIC that we use here. But we needed to bring in more equipment, including microdialysis analyzer, better brain oxygenation, temperature, cerebral blood flow, pupillometers. But I think the most important device, the most important software that we brought in was the one at the bottom that is called the sickbay data acquisition system that is was patented by a company called Medical Informatics that is from Houston, and that's where my connection comes since I started working with them when I was in Houston prior to moving back to Baltimore. And this all started from this work that Craig Russin, who was the PI on this article. Craig is actually the person who created sickbay, PhD in high frequency data analytics. And so he was working at Texas Children's Hospital with the cardiovascular surgeons and the intensivists at Texas Children's to trying to come up with predictors of trying to estimate why children that were born with single ventricles, meaning parallel circulations, were having a very high cardiac arrest rate and a high mortality rate. So Texas Children is a national referral center for children that are born with single ventricles. So, and they go through different phases, surgical phases. So between phase 1 and phase 2, those children were having a high mortality rate. So that's when Craig came in, and then he analyzed the problem with the data that was coming out of the devices was not being integrated and was not being time-stamped. So he worked on that and then worked on the analytics piece. And then he was able to determine that only 3 variables were impacting on that prediction. One of them was continuous heart rate monitoring, EKG, respiratory rate, and oxygen saturation, but they had to be continuous. And so then he was able to come up with, using logistic regression modeling, that there was a group of kids that looked very similar that had that pattern. And then when he plotted that over time, he was able to determine that there was a threshold that was crossed at 1.5 hours prior to the arrest. So 0 here means our cardiac arrest. So minus 1.5 hours, that's when those kids reached that threshold beyond which they will all look the same in the sense that they will arrest, right? So then he called that the risk index. I thought it was truly amazing that he was able to do that. So they went back to the bedside, applied the index as a code alert. And so the team was alerted to go and see those kids prior to the event, and they were able to intervene. They found they corrected whatever needed to be corrected. And the follow-up to that was that the code rates went down from over 50% down to single digits. So I was really, really impressed. And I said, well, if it's true, it's really true, can we apply this to adults? And that's when we went to the database that Claudia Robertson at the Bentop Hospital in Houston has collected over the years with patients with severe traumatic brain injury. And we defined this intracranial crisis. Of course, it was an artificial definition that we created, but we were able to show that, yes, just like in children with single ventricles, in adults with severe TBI, we could also predict, when those adults were going to go into that crisis mode. And we could do that at least 35 minutes prior to that happening. So this gave us momentum to move forward and say, well, okay, maybe we're up to something with this device. And so then Craig went ahead and patented it, and now they have FDA approval to do clinical work and to do one of the advantages of sickbay is that it's agnostic, right? So it means you can actually pretty much interface any device out there as long as you have an output, and all of them have an output. Right? So this is my thought at the beginning. I thought maybe we're going to have something simpler like this drawing here, where you're going to have all the devices and monitors that we have available, then connect them using SIGBay and move on to the analytics piece once the data have collected. And you can also see that here in this other cartoon. I think you could probably appreciate it more, but obviously this cartoon includes other signals like ventilator data, for example, EEG data that we also needed to bring in. What we mean by ventilator data, we mean all ventilator data, meaning not only the waveforms, but everything else of the indices that are derived from ventilator data, oxygen consumption, CO2 production, everything that the ventilators are able to do. And then obviously bring in all the data from the electronic health records, either everything that is quantified or is recorded, or if it's written, then we have to develop natural language processing programs to bring those data in. And then once all data is all collected and stored, then that's when we can start using the analysis for the clinical projects or for research analytics, right? But of course, it is more complicated. As you can see here, this is a real way that we have set everything up. You know, this is not a cartoon. This is more like the reality of what happened. So we have to house all that data in a secure database. So that we needed to build that and make sure that our regulatory groups, Hopkins, were satisfied with how secure the data was. And then, of course, we needed to bring everything that we could bring into SIGBay, then interface it, and eventually save it and timestamp it, right? And then we had to work with our collaborators from SIGBay, with Craig Rossin, to create tools that would allow us also to bring the data back from the database in a way that could be workable and understandable, and then be able to use that in a platform that we could work with, like using Python or Cruncher, or one of those platforms, so that we could do the analysis. There was another challenge, and one of them was obviously we had so many devices that we needed to create a way to make it easier for those to be put somewhere that it could be easier to get into SIGBay. And for that, we purchased the capsule neurons. It's also obviously we worked with this company to actually bring the devices to the site. So we installed them in every single bed, and we have 6 extra outputs here. That means that we have 6 extra devices that we can hook up and bring the data to SIGBay. So what we have so far is that, yes, we have a database storage that is safe, at least as far as we can tell, and based on meeting all the requirements from the Johns Hopkins School of Medicine. And what we're streaming per bed is about between 1 kilobytes to 10 kilobytes per second. And so what that means is our database is over 10 terabytes. So we do over 10 terabytes of data annually. So we certainly exceeded that every year that we've collected the data. And of course, we also have a specific publications committee, to make sure that everybody knows what they're supposed to be doing with their data and how they're going to analyze it. So there's no conflicts in there. And right now we're using it for clinical purposes and for research purposes, as I mentioned before. From clinical purposes, of course, we're using it for alarm management, for monitoring, for quality improvement projects, for patient safety and patient flow. And for research, of course, we're building our predictive algorithms and using computational programming and deep learning to try to our answers, right? This is our team. So it requires a lot of people from different areas and different fields working together to build this. So I'm the director of the program, and my colleague Robert Stevens is the associate director. And so we have the scientific leads here, Robert Stevens and then Vishang Shah and Rohan Mathur helping us there. They are all neurointensivists with different backgrounds, some of them engineering, some of them. And then Stan Anderson is a neurosurgeon. Elizabeth Zink is a nurse investigator and clinical nurse lead. And then Eva Ritzel is from neurophysiology as well, with a background in engineering. And then we have data managers. Peter Zijic is a software engineer, and Sylvia Caldila is a nurse, also a research nurse. And from the data scientist piece, we have Han Kim, who is a biomedical engineer. And from the biostatistical side, we have Jason Lee that works at the Applied Physics Lab. And we also have a health economist, Seema Kakkar, who got a PhD in health economics, and she's now in medical school. So then let me show you just an example of all the data that needs to be queried depending on a specific project. But in this particular instance, we're trying to come up with precision and predictions of who among all the patients that undergo elective craniotomy actually need to come to the neuro ICU, right? So currently, all of them do. All of them are automatically sent from the operating room to the neuro ICU for further management and care, at least for the first 24 hours. And what we have hypothesized is that not all of them need to come. And so that in order for that, then we need to classify the patients that do need ICU and the ones that do not need ICU, that perhaps may need intermediate care or regular floor care. So as you can see, the complexity of the data is significant because you have to keep in mind that there's different time zones that you have to collect data from. One of them is a preoperative area. Perhaps there is something with the imaging that could be predictive of need for ICU, or something related to the overall general aspect of the patient. Let's say maybe it's a patient who is very frail, or a patient that has a very low functional score, and therefore that would be a patient that would need to come to the ICU. Or perhaps there was something that happened in the OR, intraoperative, that changed everything. Maybe the patient started bleeding, or there was a complication during the procedure. Something changed that made that patient more likely to come to the ICU. And then when we look at the postoperative, then we also have to look at those first few hours of the postoperative care as well, as to what's actually happening to that patient. Maybe there was too much fluids that was given to the patient, or maybe the ventilation was not given properly. And for that, of course, we need to collect even more data, not only from the physiologic time series, but also the clinical notes, right? So we need to use, as I mentioned before, natural language processing when it's text that we're dealing with. So, and these are all these other areas that we're also tapping into because we need their help, right? So we have, I mentioned already, the Applied Physics Laboratory at Hopkins, the School of Medicine, the School of Nursing, the Technology Innovation Center, the Institute for Clinical and Translational Research, and the Bloomberg School of Public Health. I recognize that, in many ways we're sort of lucky in the sense that we have a lot of resources available to us at the Johns Hopkins University, and this may not be available to everyone in other places and in other countries. But I think one of the other things that we're trying to do with this project is also trying to first answer all these questions and then make it as simplified as much as we can, with a high degree of prediction, so that this could be applied somewhere else, right? So to conclude, I think what we're trying to tell you is that we're dealing here with big data, right? So, and big data include great variety and high velocity, right? And with big data, we have significant challenges that we have to overcome, because we have to make sure that the veracity of the data is great, that we know how to deal with the workflow, that we have an excellent computational modeling, that we also are able to extract meaningful information, and more importantly, that we're also willing to share our data, because data sharing, I think, is important, right? We also need to do that. Well, we'll talk in the next slide about the challenges for that. But I think we have to think of modern industry pioneers as examples, right? And the best examples are probably Google and Amazon, right? So I think we're all familiar with that, the general public, right? We know that when we go to do a Google search, or to go into the Amazon webpage, we start getting extra information that we didn't ask for, right? So like maybe there is that blouse or that nice shirt that is coming up for us for Amazon, telling us that we should buy that. And the question is, why are we getting that information if I didn't ask for it? Well, because they're learning from our buying habits, right? Or from our web surfing habits. So what these companies are doing even though the data that they have is messy and incomplete, they're trying to tackle our consumer data. So it's fairly homogeneous to come up with those solutions. So I think in a way we need to do something like this, because the question is whether we need to curate the data and make it perfect, or we are going to allow some degree of messiness and incompleteness so that we can actually have very large data, large databases, combine them, and yes, accept the messiness and the incompleteness to tackle the questions that were up. So obviously the future steps are, one is to develop multi-center collaborations so that we can build bigger databases. Then we have to combine the predictive algorithms. So several institutions have already come up with some algorithms. So it might be better if we just combine them. Maybe we can see what parts of these other algorithms we can use and combine them with our algorithms. Then analyze multiple and cumulative sources of data. And then once we have those artificial intelligence algorithms that are mostly derived from retrospective analyses, right? Then we have to validate them in the multi-centered way, and then prospectively, right? And the last piece will be to do a randomized controlled trial where we investigate their impact on clinical outcome, right? I mentioned in the prior slide that one of our biggest challenges is data sharing, right? So this is driven by not only by institutional policies. Maybe my institution does not want me to share the data with Harvard or with Columbia University, but there is also interdisciplinary politics, right? So maybe the traumatic brain injury group may not want us to share the data with some other group, so that's also important to keep in mind. And lastly, we need to be very mindful that what we're trying to do with all these projects is trying to emulate the human intelligence, right? So that's what we're trying to do through data-driven analytics, and to improve it, right? To make it even better, right? So obviously what that means is that because of that, then we may also reflect the biases present in our collective conscience. By that I mean, we need to work really hard to ensure that all these artificial intelligence algorithms incorporate standards to ensure fairness and health equity. Meaning we have to make sure that we take into account biases towards sex, ethnicity, race, socioeconomic status. All that has to be built into these algorithms. Otherwise, we're going to emulate what we humans have evolved to do over hundreds and thousands of years. So with that, I think I'll stop, and thank you very much. And I apologize for not being live. I had a family emergency that I had to tend to, and so my apologies. But thank you again for your kind invitation. It's been an honor for me. Thank you.